Today, we're diving into the tiny molecular machines that basically run the show when it comes to life itself. We're talking about proteins. You know, from the antibodies fighting off viruses to the enzymes that digest your lunch, proteins are the microscopic architects and engineers that make everything in our bodies work.
But you know, our journey into the world of proteins doesn't start in some high-tech lab with a complex formula. Nope. It actually starts with a messy workbench, a happy accident, and a scientist who was really paying attention.
And that scientist was Alexander Fleming. He's talking, of course, about his accidental discovery of penicillin. The story goes he came back from vacation, looked at his petri dishes, and noticed that some mold, penicillium, had contaminated one of them. And here's the crazy part. Everywhere the mold was growing, the bacteria around it were dead. Just like that, he had stumbled upon the world's very first antibiotic.
What Fleming had found was a molecule that was basically a master of chemical warfare. The genius of penicillin is all in its shape. It's a perfect mimic of a little building block that bacteria need to build their cell walls. So it sneaks in, finds a critical bacterial enzyme, and just jams it up. It stops construction cold. And without that strong cell wall, the bacterium gets weak, it bursts, and it dies. Now, to really appreciate how clever this trick is, we first need to understand exactly what penicillin is attacking: proteins.
So let's zoom all the way out and look at the fundamental building blocks of every single protein in existence. The best way to think about it is like a 20-letter alphabet that's used to write the entire story of life.
Every single letter in this biological alphabet is an amino acid, and they all share the same basic backbone: a central carbon, an amino group, and an acid group. But the magic, the thing that makes each one of them unique and gives it its own special personality, is that fourth part, the side chain, which we just call the R group.
And it's truly wild how much of a difference a tiny change to that R group can make. So take glycine. Its R group is just a single hydrogen atom. That's it. This makes it super small and flexible. But if you swap out that hydrogen for a slightly bigger methyl group, you get alanine. Still small, but it has a completely different character. Every one of the 20 amino acids has a unique R group, and that R group is what decides its size, its charge, and exactly how it's going to play with all the other molecules.
Okay, now for a really cool chemical twist. Most amino acids are what we call chiral. It just means they exist as mirror images of each other, kind of like your left and right hands. They're the same, but you can't superimpose them. Now, for reasons we don't totally understand, life on Earth almost exclusively uses the left-handed or L form of amino acids. But bacteria, they came up with a clever defense. They build their cell walls using the right-handed or D form. This is brilliant because most of our enzymes, the ones that would break them down, are built to recognize only the L form. It's an amazing bit of chemical camouflage.
All right, so we have our 20 letters. The next obvious question is, how do you link them together? How do you form the words and sentences that make up a protein?
Well, they do it with something called a peptide bond. It's pretty straightforward. Two amino acids line up, a little molecule of water pops out, and a really strong covalent bond just snaps into place between them. You do that over and over and over again, thousands of times, and you get a long chain called a polypeptide.
But, and this is a really big but, this is not just any old chemical link. The peptide bond has a secret, a special property that is absolutely fundamental to how proteins get their shape and do their jobs.
So here's the secret. The peptide bond isn't really a single bond, and it isn't really a double bond. It's kind of a hybrid of the two. And because of that, it's really rigid and flat. It can't just rotate and spin freely. Think of it like a hinge that's kind of stuck, only allowing for movement at specific angles. This inflexibility is actually the very first rule that starts to force that long floppy chain of amino acids to begin folding into a very specific shape.
You know, one source put it in a way I just love. It said that if proteins were just long chains of carbon, they'd be as boring and inert as a piece of plastic. To make them useful, biology has to spice up the inertness. And it does that by sprinkling nitrogen and oxygen atoms into the protein's backbone. These atoms are, you could say, electron-hungry, which creates little areas of positive and negative charge. And it's that polarity that allows for all the interesting chemistry that life depends on.
So how do all these little chemical rules, the unique personalities of the R groups and that special, rigid peptide bond, come together? Well, they all work in concert, like a tiny orchestra, to guide the protein from being just a simple string into a complex, three-dimensional machine.
The first level of this is super simple. The primary structure is just the order of the amino acids. That's it. It's the unique linear sequence of the letters in the chain, a list of ingredients, if you will.
But that chain doesn't stay straight for long. Next, it starts to fold up into these little local repeating patterns. This is the secondary structure, and it's held together by a whole network of hydrogen bonds that form along the polypeptide backbone.
Now, the two most common patterns you'll see are the alpha helix, which is this right-handed coil that looks a lot like a spring, and the beta sheet, which is where different parts of the chain lie side by side, kind of like a pleated ribbon. In that alpha helix, all the R groups stick out from the coil, but in the beta sheet, they point up and down, alternating along the chain. These little local structures are the first step in building the protein's final complex architecture.
And all of this leads us to the grand finale, the tertiary structure. This is the whole shebang. It's the overall, final, three-dimensional shape of the entire protein chain. All those little helices and sheets fold up on each other, guided by all those different interactions between the R groups, creating a one-of-a-kind functional structure.
Okay, so now let's bring this all the way back to that moldy petri dish, because this is where we see how all these fundamental chemical rules become, quite literally, a matter of life and death.
And here's the genius of it all. Remember how bacteria use those right-handed D-amino acids as a defense? Well, it turns out that penicillin's molecular shape is a near-perfect imposter of a little peptide D-alanine that the bacterial enzyme is looking for. So the enzyme gets completely fooled. It binds to the penicillin thinking it's the real thing and gets permanently stuck. It's disabled. And with no enzyme, there's no cell wall. And with no cell wall, there's no bacteria. It is a stunningly beautiful example of molecular mimicry.
So what are the big takeaways here? Well, we've seen that proteins are built from a simple 20-letter alphabet of amino acids, where each letter has its own unique properties. We've seen that they're linked by a special rigid bond that forces the whole chain to start folding. And all of this leads to the most important rule of all. A protein is forced to fold into a very precise 3D shape, and it is that shape, and only that shape, that determines its function.
Which brings us to a final, really powerful question. If we truly understand these fundamental chemical rules that life itself is built on, what does that mean for us? It means we can start to use them ourselves. We can design new drugs that, just like penicillin, target a specific protein shape. We can even start to engineer entirely new proteins to do things we've only dreamed of. If chemistry is destiny, then the future is something we can actually help write.